So I'd love, great honour is to introduce Anna Santos, who's going to present some of the work that she carried out with Barry Johnson. Over to you, Anna.
Thank you very much. Thank you, the organisers, for giving me the opportunity to talk to you today. I could put my camera on as well if I can. Yeah, you'll probably be able to see me. I can't see myself. So thank you for giving me the opportunity to talk to you today. And I would also like to thank this great group of people who made this work possible. Let me just see if I can get my laser pointer here. And also this work is being carried out under the Crocodile project, the Horizon 2020 funded, which looks at recovery of cobalt from two different sources, the raw materials and then from batteries. Before I start talking about chromium inhibition in bioprocessing of limonitic laterites, I want to give you a little bit of a background story first. It's starting off with what are laterite ores. They are supergenes ore bodies originated from chemical and mechanical weathering processes, mostly in tropical and subtropical areas. But you can also find laterite deposits in areas such as the Mediterranean, the northwest coast of the US, also Russia and Kazakhstan. Laterite ore deposits, they follow a similar weathering profile, which is generally divided in three main layers. At the bottom, you have clay silicates with dominant nickel smectites, above which there is a saprolitic layer, rich in silicates as well, also rich in magnesium nickel hydrosilicates. On top of that, there is an oxide zone of limonitic laterite consisting of iron oxides dominated by goethite and manganese oxides such as asbolane and lithiophyllite. In the photo on the right hand side, you can clearly see the difference, the different layers of the laterite deposit. This one is a photograph from Kaldak mine in Turkey, and you see here a green saprolite zone at the bottom, covered by a thick layer of the limonite zone, which is the goethite rich zone, and on top of that there is a white silica rich cap. Laterite mining started in the 1870s in New Caledonia for nickel production using laterite deposits. However, with the discovery of sulfide deposits containing nickel and copper in Canada, shifted its production to sulfide ores. And one of the reasons for that is that laterite processing is highly energy demanding, but not only that, it's also it's a very complex and very heterogeneous type of ore, and that can make the downstream processing very costly. Interested to know that laterite accounts for around 70% of the world's accessible nickel reserves, but nickel production from laterites is about 42 to 46% nowadays. Saprolite is the most processed, is mostly processed using pyrometallurgy with the production of ferronickel. And while we do have hydrometallurgical techniques to process limonites, for example, the Chromium process, the HPAL, the high pressure acid leaching, and heap leaching, the associated costs and engineering complexity means that the limonite layer of a laterite deposit is usually discarded as mining waste. Talking about nickel and cobalt deportment within lateritic ores, nickel is typically associated with iron 3 minerals, where cobalt is usually associated with manganese 4 minerals. You can also find nickel in the manganese 4 minerals, for example, asbolane. Other types of laterite ores can also have significant concentrations of copper, scandium, and in some cases rare earth elements. Laterite contributes to 20 to 30% of total global supply of cobalt. Cobalt that is otherwise obtained as a co-product from copper and nickel sulfide ore processing, with the one exception in Morocco with the Bouaza mine, which the target metal is cobalt. We've seen recently, even more recently, the exponential increase in demand for metals such as cobalt and nickel, especially due to their increased use in high-tech materials. The one that we've all heard very recently with the COP26 is the use of the rechargeable batteries for electric cars, but also in superalloys and catalysts. And although processing limonite can be a major solution to help to meet the demand of nickel and also help with the supply of cobalt, scandium, and other elements, there is still lack of a novel and sustainable robust processing route that require less energy and reagent inputs and produce non-polluting residues. And the biohydrometallurgy can help a little bit with that, and I'm going to show you how. The term sulfur-enhanced bioleaching has been used to describe the microbially accelerated dissolution of minerals carried out under both oxidizing and reducing conditions. In the case of oxidized ores, we need to apply reductive bioleaching as opposed to oxidative bioleaching. This technique uses bacteria that couple the oxidation of elemental sulfur to the reduction of iron 3 to iron 2. And the bacteria that reduce the iron 3, mostly strains of acidified also oxidize iron 2 when oxygen is available. So these processes usually being carried out under anaerobic conditions. And it is divided into main steps. The first step is the biomass growth, where oxygen and sulfur are provided, and the sulfur is inoculated, and you wait for the biomass to grow. Then you halt the aeration, and you start pumping oxygen-free nitrogen to create reductive conditions, and then you add the limonite ore. The most important reactions happening here are the reduction of the goethite phase, which is coupled to the oxidation of sulfur by bacteria, and with the production of ferrocene and release of nickel. The second most important reaction in the reductive bioleaching is the reduction of the asbolane phase, which is this mineral here, that is attacked by ferrocene and then releases nickel, cobalt, manganese, and ferric ion. The ferric ion produced here is then reduced by bacteria. So in every slide that I showed you about, with all the results we've got, we are looking for low redox potential. So I want you to keep that in mind, because I'm going to talk about that a bit later. All right, so moving on to the materials we've tested. Within the Crocodile project, we received 2 sets of samples, one of them from Greece. We received 5 samples. 2 of them were processing residues. This mine in Greece is operated by Larco, and they use parametallurgy to produce ferronickel. So the filter dust and the slag are the residues. And interesting to know that from all of the samples that they sent us, the highest concentration of cobalt was in the filter dust. So they are losing most of the cobalt when they are processing the nickel for ferronickel production. So it's something that we looked at and thought this is very interesting. I'm not going to go into detail. You can access the paper if you want to know more about that. What you need to know now is that all these materials, they were amenable to bioleaching, and that the reductive conditions, they were maintained throughout the experiment. They were all amenable to different extents, of course, and you can find more about it if you are interested. What I am going to focus today is the samples that we received from our colleagues at BRGM. three samples from New Caledonia, but I'm only going to talk about two, NEC1 and NEC2, both from the same deposit, Penamax. NEC1 was a core drill sample, and the NEC2 is a bulk sample. What I want you to see here is that, where's my thingy? There. I want to highlight is just the difference between these limonites. And you see here that limonite NEC2 contains a lot more iron, a lot more chromium, manganese, nickel, and cobalt than NEC1. This NEC2 below 50 here, I will talk about it a bit later. And you see that, and that's gonna explain a little bit more later. Again, the concentration of target metals being slightly higher than those at NEC1. All of these limonites, including the Greek ones, they were all screened first, the preliminary experiment, the bioleaching experiment. They were all carried out the same way, and using oxygen-free nitrogen with slight changes in pH, and they all, the pulp density ranged from 1 to 15%. And they were inoculated either with a mesophilic consortium containing acidithiobacilli and a moderate thermophilic consortium including Acidithiobacillus caldus and Acidianus rarelii and sulfidivorans. So we've carried out lots of different experiments, and now is when I start telling you a story on how we started, how we identified this chromium issue. The first experiment we did with the New Caledonia samples. We used the mesophilic consortium, pH 1.5, 35 degrees, and 10% pulp density. What I told you before is that we are always looking at low redox potential. So in the first experiment with this new sample from New Caledonia, once we added the limonite to the bioreactor, the redox potential went up and was kept high throughout the whole experiment. And that also shows here as the open symbols are ferrous iron and the closed symbols are total iron. So all of the iron there was present as iron 3, which is not really what we want. And we investigated a bit further to see what the problem was. And I noticed that looking under the microscope and also doing a field analysis after these experiments, that the microbial activity was completely inhibited, and that happened after a few days. You could look under the microscope, and the cells were gone; they completely disappeared. And then we started thinking, how is that? Why is that happening? And a potential cause here is the presence of oxygen ions in these samples, limonite samples. And to basically support our idea of the chromium could be the issue, we found there's this group in 2009 who found that 25% of total chromium in a limonite deposit, also from New Caledonia, 25% was present as chromium-6, and that in some localized points, this concentration could go up to 33%. And they postulated that this arose within the limonite from the oxidation of chromium-3 by manganese oxyhydroxides, and suggested that the chromium-3 was released from the iron oxides located near manganese oxyhydroxide boundaries, oxidized chromium-3 to chromium-6 before this chromium-6 was again incorporated onto the surface of surrounding iron oxides. And if you look at redox potential of the couple of the MnO2 and manganese 2 couple, it is strongly electropositive, being able to catalyze the oxidation of chromium-3 to chromium-6. So here, just to summarize what I've talked until now, is that if we don't have reductive conditions, if our leaching is an oxidative leaching, the presence of ferric iron and chromate, chromium-6, our bacteria are not happy. But if we do create reductive conditions, and then that ferrous ion will reduce chromium 6 to chromium 3, our bacteria will be happy. So then we had to find a way to do that, to make sure that we had ferrous ion in the system. So we're using the same limonite. We did a second experiment using the same conditions, the same consortium, but then in this case, instead of adding the 10% pulp density all in once, we added a little bit at a time. So instead of giving them a full meal, we just gave them snacks every now and again to see if that would help. And it did. You can see here that the redox potential goes down, and every time you added a bit more of limonite, it would go up slightly, but then back down again, and then up slightly, and then back down again. And the metal recovery really improved when we did these incremental increase, with recovering over 90% of cobalt using this approach. The problem here is that it took 50 days for that to happen, and industrially that's never gonna work. So we then move on to New Caledonia 2, which is the one with higher concentration of cobalt, and we tweaked it a bit. So instead of using the mesophilic consortium, we then used the moderate thermophilics, just trying to speed things up. And we kept the incremental increase, so just give them a little bit at a time. And just to see if that would help it even more, we added 2 millimolar ferrous ion into the reactor prior to the bioleaching. And the rationale here was, once the limonite was added, the chromium 6 released into solution would then be reduced straight away by the ferrous ion that was already in the system, without having to wait for the microbes to reduce that ferric ion. So what happened here in the beginning was pretty much the same. So the redox goes down and then it goes up again once another addition is carried out. But at this stage here, when you reach the total of 10% pulp density, the redox did not go down. So we went up and it stayed up, and which was not really what we wanted. But still, the metal recovery wasn't too bad. We're still recovering over 70% of the cobalt. So then we tried it again, and the difference with this experiment is that we did the additions of limonite even in smaller portions. So we started at 0.5%, and then we made our way up to 15%. And we added more ferrous ion in the beginning as well, because this limonite contained more chromium than NEC1. So then we thought, yeah, let's just add it a bit more and see if that helps, and if it will go all the way up to 15% and not stop at 10. And here again, redox went down. There is the little fluctuation every time I added a bit more, and after we completed the 15%, the reductive conditions were kept until the end of the experiment. And here we are talking about 18 days instead of 50. So that is already reduced massively the leaching time. Again, our metal recovery wasn't too bad. Cobalt over 70% and manganese around 80%. And so then, we stopped and thought about how much ferrous ion do you actually need to do this experiment and get two things done. First, for all the chromium 6 to be reduced to chromium 3, and for all the ferrous ion to reduce the asbolane phase, because here again we are looking for cobalt. So then we did a fifth experiment. But in this case, it was an abiotic experiment, because we just wanted to see how much ferrous ion we would need. And because we didn't have to really care about the bacteria being intoxicated, 15% all in at pH 1.5 and 50 degrees. And what we did in this case was adding instead of adding limonite, we added ferrocene to see if how much ferrocene we would need for these two purposes. And in total, we needed 30 millimolar, because you can see here that it is when ferrocene is added and then is completely consumed and then is added again, and then it was consumed to a certain extent, and then it kept the same concentration. So then we thought, so that's it, that's all gone. And then we did the metal analysis, and again our 70% cobalt is always there, really regardless of the conditions. What is interesting about experiment 5 is that again the time scale is very, very promising. We are now talking about hours, not 15 days. So we went from 50 days to 18 to about 3 days now, which we were very happy about this part. And where's my next slide? There we go. So now I'm going to move from leachate analysis, and I'm going to talk to you a little bit about the leached residues, the residue analysis. And here the XRD patterns for NC1 showed that the chloride peak, which is this one for the raw limonite, it was completely disappeared or was greatly reduced, and that the goethite, which is the iron oxide, was not, it didn't change almost at all, which does agree with the chemical analysis, because we didn't get much iron or nickel recovered from these experiments. And pretty much the same happened with NC2. And the only phase that appeared in these XRD patterns is the sulfur, but this is the sulfur that was added at the beginning for the biomass growth. We also did some SEM analysis, and this one, the blue represents the ion distribution, red the manganese, chromium is the green, and silicon is the yellow. This is the raw limonite, and this is the two leached residues. Here is just to show the sulfur distribution. So you can see that the sulfur that we added for the biomass is all over the place on the surface of the limonite. And here for NC2, iron is green, manganese is red, and chromium is blue. You can see here that all the red, there's a beautiful red vein there, is completely, there's almost no manganese left there, which again agrees with the chemical analysis, because we got a high manganese recovery from these experiments. But at this point, we still didn't know if we did really have chromium-6 in our samples. We knew that there was another New Caledonia sample that had chromium-6. So then we carried out some analysis, zane analysis, and with this spectra here, you can see that there are revealed two distinctive components, one at 5990 and another one at 5993. And this one is an excellent indicator of presence of chromium-6. And the intensity represents it represents the concentration of chromium-6. LAR4 is one of the Greek limonites, and you can see that it's almost nothing compared to the NC2 and NC1 limonites. And that's interesting, because we didn't have this problem when we processed the Greek limonites, and here is why. And again, it supports our hypothesis that the chromium-6 is the problem. So we know that there is chromium-6 in the limonite, but we wanted to see if there was some chromium-6 also in the leachate once after the leaching experiment. So a really, really nice and quick experiment that we did, an abiotic experiment, where we mixed 20% of the NC2 at a very, very acidic liquor containing 100 millimolars of either ferrosulfate or magnesium sulfate, and we incubated that 2 days. And then these are the analysis. And what I want to highlight here for you is that the redox potential for the magnesium extract is very high. Which
you've got 5 more minutes.
Okay, I'm almost there. The redox potential and with the magnesium extract, it was very high, and around 680 when we used the ferrous sulfate extract. Here, the total concentration of chromium for the manganese extract and the iron extract. And interesting that the chromium 6 here was not detectable when with the ferrous iron extract, which is expected because we wanted the ferrous iron to reduce that chromium 6. And you can see that it was detected in the magnesium sulfate. Okay, this was all abiotic. What we wanted to do now was to try to oxidize biologically the remaining ferrous iron in these two leachates for our downstream processing. But then we reached a conundrum. We did a toxicity test. We wanted to see if acidophilic iron oxidizing bacteria were able to oxidize the remaining ferrous iron in these two leachates. What we found is that the magnesium extract, we tried with these leachates the way they are, no oxidation whatsoever. So then we tried diluting it serial dilutions, and with the magnesium extract, diluting 10-fold or more, iron oxidation occurred. But the ferrous sulfate extract, we had to dilute 100-fold or more to see any signs of iron oxidation. And that's the big question for us, and it still is, because we thought at first that adding ferrous iron, we were helping the process, but then for the downstream processing that we need that remaining ferrous iron to be oxidized, it's still not working, still very toxic. And I will finish that. This is my last slide. So what do we know so far is that the reductive bioleaching of limonitic laterites may be inhibited by chromium 6, as we could see with the two different sets of samples. The inhibition can be partially or completely eliminated by adding ferrous iron at the beginning of the bioleaching, or by incrementally increasing the pulp density of the reactor. Acidic ferrous iron extracts of the limonites, they can still be highly biotoxic, and we still don't know why that's happening, but there are some more work on that. And last but not least, the potential impact of this work in the development of reductive bioprocessing for limonites is very, very significant, it's highly significant. Mainly because the key reaction in the process is the microbial ferric ion reduction. And if that doesn't happen, if the reduction of ferrous ion does not occur, that the metals are only being leached by acid, which is not very effective. And I thank you again for listening.